Alright, so now we move on to the next technique. Electrophoresis is a separation technique in which charged molecules could be separated from one another by using an electric field. So when we apply a potential difference that results in the flow of electrical current, so the charged molecules are going to migrate in that electric field. The substances which are positively charged will migrate towards the negative terminal, and the ones which are negatively charged will migrate towards the positive terminal. So electrophoresis is a separation technique which separates molecules on the basis of their charge. Biomolecules, they have ionizable groups on them. A variety of different types of biomolecules, including nucleic acids, proteins, and peptides, carbohydrates, they contain charge groups on them. A few examples of the charge groups include amino group, carboxyl, phosphate, and some other groups. Some lipids also contain charges and could be separated by means of electrophoresis. Most lipids are nonpolar and they are not charged. However, a few lipids, like phospholipids, they contain charges on them and could be separated through electrophoresis as well. Mostly, we separate proteins and nucleic acids by the process of electrophoresis. Nonetheless, the biomolecules, they contain many different types of functional groups on them which can bear charge. And based upon these functional groups which impart charges to these biomolecules, separation could be done by using electrophoresis. So the ionization of the groups is dependent upon pH. At a given pH, the groups present on the biomolecules, they may be ionized and they may be charged as well. But at some other pH, the ionization of the groups may be different. So the charge on a given biomolecule is dependent upon the pH. At any given pH, the biomolecules can have a positive or a negative charge. Proteins, for example, they at a certain pH, the proteins, they may be containing more positive charges as compared to the negative charges. So the net charge on the protein would be positive. If a protein contains more negatively charged amino acids on it as compared to positive charges, so the net charge on the protein would be would be negative. Another condition would be that at a given pH, the number of positive charges of a protein would become equal to the number of negative charges. So the net charge on the protein would become 0. Such a pH where the net charge on a protein is 0 is called as the isoelectric point of that protein. Migration during electrophoresis is dependent upon the electromotive force. So when the molecules are placed in a gel matrix, they are going to migrate under the influence of the electric field, and the molecules are going to migrate towards the oppositely charged terminal. So migration of the molecules within a gel during electrophoresis is dependent upon their charge to mass ratio. So charge to mass ratio is an important factor in governing the rate of migration of molecules in a gel. The gel matrix is a solid matrix. It is a porous matrix which provides channels for the movement of the biomolecules for separation. For example, nucleic acids, when are separated, they all have the same charge to mass ratio. So when subjected to a gel matrix and electric field is applied, so the molecules are going to migrate through the gel matrix. Those molecules which are smaller, they can easily move through the channels of the gel and their migration rate will be more. As compared to the molecules which are larger in size, and hence it would be difficult for them to migrate through the pores of the gel. So the migration rate of the large molecules would be relatively less. Based upon this difference in the migration rate, separation of these molecules will be done. Those molecules which are smaller can migrate easily and therefore they will travel greater distance. The molecules which are larger in size will migrate at a slow rate and hence they will travel less distance within the gel. So based upon the relative distance they travel, these molecules will be separated from one another. The commonly used gel matrices include agarose and polyacrylamide. Proteins are separated by using polyacrylamide gels, whereas nucleic acids, including DNA and RNA, they could be separated by both agarose and polyacrylamide gels. Large nucleic acids, like hundreds to thousands of nucleotides, they could be separated on agarose gel because the pore size offered by agarose gel is larger. However, small nucleotides like oligonucleotides or like tens to hundreds of nucleotides, they cannot be effectively separated on agarose gels. So small nucleic acids could be separated on polyacrylamide gels as well. The gel is a porous matrix, as you can see here in this electron micrograph. So there are these channels or pores present within the gel. So these pores act like a sieve. So through the sieving property of the gel, the molecules are separated. Those molecules which are smaller, they would be able to easily migrate through these pores, whereas the ones which are larger, it will be difficult for them to migrate through the pores of the gel. So the gel offers resistance to movement, and this impedance which is offered by the gel is the basis of the sieving property of the gel. So the gel has these pores in it, so the pores act like a sieve, and based upon the pore size, the migration rate of molecules are determined. This leads to a difference in the migration rates, and hence molecules, they could be separated from one another. Gel concentration is one important factor for determining the pore size. When the concentration of the gel is more, the pore size is smaller. So the pore size of the gel is inversely proportional to the concentration. So a gel having higher concentration will have a low pore size, and hence the migration rate will be low. As opposed to this example, a gel having low concentration will have larger pore size, and hence the migration rate of molecules in that gel would also be high. Electrophoresis could be done in native as well as denaturing condition. The native or the non-denaturing condition is the one in which the biomolecules, they remain in their original physiological structure. They are not denatured. The denaturing gels are run when the biomolecules, they undergo denaturation by using some chemical agent. Native and denaturing gels can be run for both nucleic acids and proteins. Most of the time when we separate nucleic acids, we carry out native electrophoresis by using agarose gels. During native electrophoresis, DNA maintains its double stranded structure, and this is one of the commonly used mechanisms to measure the mass of DNA molecules which are being separated. DNA is a double stranded structure, and if through some action we separate the two strands, so that is called as melting or denaturation. One mechanism to melt or denature DNA is to heat it. Upon heating, the two strands of DNA separate. Another way is to add chemical denaturants like alkali. So addition of alkalis results in separation of the two strands of DNA, Hence, DNA becomes denatured. It becomes single-stranded. Denaturing electrophoresis is also used for some applications. For example, when DNA hybridization has to be carried out, so DNA must be first denatured and converted into single-stranded form. Proteins can also be separated through electrophoresis under both native and denaturing conditions. Under native conditions, every protein will have a particular charge to mass ratio. So when the properties of the native protein are to be studied, for example, interaction of protein with other proteins, formation of larger protein complexes. So when the native properties are to be studied, or for contribution of a protein in complex formation is to be studied, then native electrophoresis is done. Denaturing electrophoresis is generally done for the separation of proteins. Under denaturing conditions, the charge to mass ratio for all proteins become equal, and hence the proteins are separated on the basis of their molecular mass. So when the mass of a protein is to be estimated, it is through denaturing electrophoresis. Also, some proteins can be made up of more than one polypeptides. They can have more than one type of subunits as well. In order to determine the composition of a protein, that how many subunits it has, how many polypeptides it has. So under those, if this is to be determined, then denaturing electrophoresis is also done. So under denaturing condition, the protein, all of the subunits are going to be separated from one another, and then they could be resolved and separated by using denaturing electrophoresis. There are several factors which affect the migration of charged substances during electrophoresis. One important factor is the net charge of the molecule. The charge, for example, in case of proteins, the charge is dependent upon the pH. At a certain pH, the protein may be having greater positive charge. At some other pH, the protein will have less positive net charge. At some other pH, the protein may be negatively charged, and the magnitude of charge may also vary. So one important factor influencing the rate of migration is the net charge of the molecule. Greater is the net charge of the molecule, more is the rate of migration. The size and the shape of the molecule is also important in this regard. Those molecules which are spherical in shape, or they have a compact shape, they can easily move through the gel matrix, and hence their migration rate will be more. For example, DNA molecules can exist in a circular or a linear form. Those DNA molecules which are circular and they're coiled, they will be more compact. So circular coiled molecules of DNA will migrate at a faster rate as compared to linear DNA. The size of the molecules is also important here. Those molecules which have a larger size, their migration rate would be slow because it would be difficult for them to migrate through the pores of the gel. Those molecules which are of smaller size, they can easily move through the pores of the gel, and hence they will travel greater distance and the migration rate will be more. The buffer which is used for electrophoresis is also important in governing migration. At a particular pH of the buffer, the molecules will have a particular typical charge. So the charge on biomolecules is dependent upon the pH, so the buffer pH is important here. If the buffer pH is such that the magnitude of charge on the molecules is more, then the rate of migration will also be increased. Not only pH of the buffer, but also buffer concentration affects the rate of migration. If the buffer concentration is more, then there are more ions present in that buffer. More ions means greater electrical conductivity. So if the concentration of a buffer is more, then the electrophoretic pull on the molecules is also greater, which results in a greater rate of migration. If the concentration of the buffer is low, then the number of ions in buffer are also low, and hence the migration rate will be decreased. The strength of the electric field is also important here. Greater is the intensity of the electric field, more will be the electrophoretic pull, and hence greater will be the migration rate. Less is the strength of the electric field, so the migration rate will decrease proportionally. The properties of the support media or the gel are also important here. That is, the pore size, the ability of the monomers to polymerize and form the gel matrix. The temperature of the operating system is also important. Increase in temperature results in increase in the electrical resistance as well. So it influences the intensity of electrical current and hence the rate of migration. This slide shows the pictures for the electrophoresis system. Here, this is a typical gel tank for agarose gel electrophoresis, which after placing the gel and filling in the buffer, it is connected to a power supply and the run could be done. Here, it shows the system for polyacrylamide gels. So this polyacrylamide gels are run in a vertical system, and this is the gel tank which contains the gel and the electrophoresis buffer in it, and it could be connected to a power supply and a run could be done. This is showing the electrophoretic chamber for agarose gels. Agarose gels are produced as horizontal slab gels, and these are the combs which are inserted in the gel to make a well. A well is an empty space in the gel where a sample could be loaded. So now we are going to talk about agarose gel electrophoresis. So agarose gels, they are made up of agarose. Agarose is a heteropolysaccharide. It contains a repeating disaccharide unit in it, which is made up of galactose and anhydrogalactose. So this repeating disaccharide unit makes the agarose molecule. Agarose is a heteropolysaccharide and it is soluble in aqueous systems. Its solubility in cold aqueous systems is low, so therefore agarose gel can be dissolved in hot buffers. The buffers which are used for agarose gel electrophoresis, they include TAE, TBE, and TPE buffers. These buffers, TAE is one of the commonly used buffer. These buffers, they include tris chloride as a mechanism to regulate pH. This provides buffering, contributes to buffering capacity. It has ions which contribute towards electrical conductivity. And then it has acetate ion. Acetate ion is contributed by adding acetic acid. So both tris and acetate, they contribute to regulating the pH and providing ions for electrical conductivity. E stands for EDTA. EDTA is a chelating agent. It binds to metal ions, positively charged metal ions, and many of the enzymes which act upon nucleic acids, they require magnesium ions as a cofactor. So addition of EDTA, which is a chelating agent, it binds to positively charged cations, so that results in sequestering and binding of the magnesium ions, so that if there are any modifying enzymes or any modifying agents present, So they will become inactive because of addition of EDTA. Also, EDTA contributes to the overall electrical conductivity. So TAE buffer contains tris base, acetic acid, and EDTA in it. TBE buffer contains boric acid instead of acetic acid. So it contains tris base, boric acid, and EDTA. Another buffer used is TPE, which contains tris base, phosphoric acid, and EDTA. So the agarose gels, they are cast in buffer, and then the buffer is added to the electrophoresis tank, and hence electrophoresis could be carried out. Now, when we add DNA to the gel, DNA is loaded into the wells. So a well is an empty space in the gel where the sample could be loaded. Now DNA samples, they are colorless and they are also aqueous solutions. The buffer which is used for electrophoresis is also an aqueous solution. So the density of the DNA is similar to the density of the electrophoresis buffer. So when we add the DNA specimen to the well contains buffer in it, so DNA is not going to settle in the well. In order to settle DNA solution in the well, the density of DNA solution should be increased. This is done by using a loading dye, which is sometimes also called as the gel loading buffer or the sample loading dye. The dye contains tris chloride in it. Tris chloride is important for regulating the pH. It contains glycerol in it. Glycerol has higher density as compared to the density of water. So when this dye is mixed with the DNA sample, by virtue of the glycerol present in it, the density of DNA sample also increases. It contains EDTA in it. EDTA is a chelating agent, and again this is to inhibit any nucleic acid modifying enzymes, because these enzymes, they use magnesium ions as a cofactor. So EDTA will sequester any cations which are present in the medium. In addition, the electrophoresis buffer, it contains colored dyes in it. The commonly used dyes include bromophenol blue and xylene cyanol. Now, the solution of DNA is colorless, so it will be difficult to monitor if the solution is going to be loaded into the gel or not. Also, when the run is in progress, it would be difficult to monitor the progress of the run because DNA solution is colorless. So it will not be possible for us to judge how much migration would have taken place. So to make it visible, these color dyes are added to the loading dye, and one important function of adding the color dyes is to make the DNA solution visible while it is being loaded onto the gel. Another important implication is that bromophenol blue, it migrates at a rate similar to the migration of about 300 base pairs DNA. So the dye is also negatively charged, and it migrates towards the positive terminal. So when DNA is migrating, it will not be visible in the gel, but bromophenol blue, which is a blue color dye, it would be visible. So we can use this as a tracking dye to notice the migration of DNA within the gel. So for example, here you see that this is the gel, and this blue color which you see over here is because of bromophenol blue. So that means the molecules of DNA, they would be somewhere here in this region. Bromophenol blue, its migration rate is similar to the migration of a 300 base pair DNA in 1% agarose gel. So therefore, when the sample is loaded, the blue color would be visible in the well, and as time passes and electrophoresis progress, the blue color of bromophenol blue is going to migrate. Hence, we would be able to see how much migration would have taken place. So, depending upon how much migration is needed, then we can switch off the power supply. Otherwise, in the absence of this blue color dye, it would be difficult to monitor the migration of the specimen in the gel. It is important to note that bromophenol blue does not color the DNA. DNA is visualized by staining with other dyes. Bromophenol blue is a tracking dye during the process to monitor the process of migration. DNA can be visualized by using fluorescent dyes. One of such dye is called as ethidium bromide. Ethidium bromide, it absorbs light in the UV range and then it emits fluorescence within visible range. So ethidium bromide, it intercalates or stacks within the double helical structure of DNA. So ethidium bromide is used to stain DNA. So it produces fluorescence, and then by monitoring fluorescence, the position of DNA in the gel could be monitored. So DNA or RNA, they both could be stained by using ethidium bromide. The detection sensitivity for double-stranded DNA by using ethidium bromide is more as compared to single-stranded DNA or RNA. Staining of DNA with ethidium bromide could be done in several ways. Ethidium bromide can be mixed with the DNA sample and the run could be done. Another way would be to add ethidium bromide in the gel while the gel is being poured, so that the entire gel contains ethidium bromide in it, and when DNA is running through the gel, it accumulates ethidium bromide in it. Another way is to run the gel without addition of ethidium bromide, and then later, after the run is complete, then the gel could be submerged in a solution of ethidium bromide. And ethidium bromide enters the gel through diffusion and binds to DNA. Later, the gel could be exposed to UV light and fluorescence could be monitored. This is how a gel would look like under UV light. So here you see the gel. The region of the gel which is producing fluorescence is appearing bright. And the region of the gel which does not contain DNA is producing very little fluorescence and hence is going to appear dark. So this is a band of DNA which is producing fluorescence because ethidium bromide is bound to it. You can see that in this lane 1 there is a certain amount of DNA present, but lane 2, 3 and 4 they contain greater fluorescence in them. So the fluorescence is proportional to the amount of DNA. One important aspect which we can study by using the electrophoresis is that we can determine the mass of DNA. The mass of DNA could be determined by using some reference. So here in this lane M, which stands for the marker, so the marker contains a series of known DNA fragments. The size of these DNA fragments is known and the markers are available from commercial suppliers. Sometimes the markers are also called as ladder. So the marker or ladder is actually a standard or a reference. So they contain multiple bands of DNA and the size of each of the band of DNA is known. Now the rate of migration, when we compare the rate of migration of a standard with the rate of migration of a DNA with unknown mass, so we can estimate the mass of this DNA molecule. For example, let us say that if this band is of 10 kilobases, this one is of 8 kilobases. So we can see that this molecule or this band of DNA is closer to 10 kilobase. So that means the size of this DNA is around 10 kilobase. So depending upon the rate of migration and then by comparing the position of a band of DNA with the standard, we can estimate the molecular mass of that DNA. This is one important parameter which we can deduce from Agarose electrophoresis, that is the size of DNA molecules. Another important parameter is the amount of DNA. Now you can see that the fluorescence intensity is increasing in this order from left to right. This is because of the reason that the amount of DNA is also increasing. Greater is the amount of DNA, more ethidium bromide will bind to it, and hence greater fluorescence will be produced. So the amount of fluorescence produced is proportional to the quantity of DNA present in a band. This can again be standardized that we can run known amounts of DNA on a gel and can monitor its fluorescence. And then we compare the fluorescence of those standards with a DNA band having unknown concentration. So when we compare the fluorescence intensity of an unknown band of DNA with the standards, so we can estimate how much fluorescence the unknown band of DNA is producing. And then by matching the fluorescence intensity with the standards, we can estimate how much amount of DNA is present in that band of DNA. So these are the two important parameters which we can deduce from agarose gel electrophoresis. That is, we can determine the molecular mass as well as we can estimate the concentration of DNA present in that band. The images of gels are recorded by using a system which is called as the gel documentation system. The gel documentation system has a UV illuminator, which is actually a UV lamp, and the gel is placed on a sheet associated with the lamp, and the lamp is switched on. So when the illuminator is switched on, it gives away UV light. Ethidium bromide present in the gel absorbs that light and produces fluorescence. There's a camera attached to the instrument. The camera records the image of the gel, and then the image is captured by a software which is installed on a computer which is connected to this camera. So in this manner, the camera takes the picture of the gel and it is recorded in the software, and then it could be manipulated, and several manipulations could be done in an automated fashion. For example, the software can accurately estimate the size of DNA. The software can also monitor the fluorescence intensity and can provide us information about the concentration of DNA in the given specimen. When the process is done for detecting RNA, so RNA molecules, they could be separated on a gel and then detection could be done. So this process is called as Nadran hybridization. A similar process could be done for detection of proteins. Proteins are detected by using suitable antibodies. So first of all, proteins are separated by using electrophoresis, and then the proteins are transferred to a membrane and then detected by using antibodies. This is called as Western blotting, or sometimes called as an immunoblot. Another important implication of electrophoresis is to study the interaction between nucleic acids and other substances. This mechanism is called as electrophoretic mobility shift assay. If a nucleic acid, let us say a double-stranded DNA, is migrating in a gel, It will migrate at a certain rate and will occupy a certain position on the gel. Now, for example, if we add a protein which binds to this DNA, then the protein will be bound to DNA and hence the size of this DNA protein complex will increase. When the size of the DNA protein complex will be more, its migration rate will become less. So the mobility of this DNA will be retarded and it will travel less distance. Now in this illustration you can see that in lane 1 there is this DNA which is going to migrate at a certain rate. This lane contains only DNA in it. So this DNA is going to migrate at a certain rate and after a definite period of time will occupy this position on the gel. Now in lane 2, this is a mixture which contains DNA and some interacting partner. If DNA was interacting with that partner, let us say a protein, then if the protein was able to bind to DNA, then it will form a complex and the size of the complex will become more. When the size of the complex will become more, its migration rate will become less. So this DNA will slow down and its migration would become less, as represented here in this lane 3. So if let us say a protein is added which binds to DNA, so the protein forms a stable complex with DNA and hence as compared to this one, the rate of migration becomes less, so the mobility of DNA is changed. So the position of the band is shifted. In this case, if the protein added is not able to interact with this DNA, so the protein does not bind to DNA and hence there is no change in the migration rate of DNA. So if one type of protein is added here and there is no change in the mobility shift, that means this protein does not bind to this DNA. However, as compared to the control, if upon addition of a protein we see a change in the mobility, a shift in the mobility of DNA, that represents that the size of this DNA has increased. The increase in size is because of the bound protein to this DNA. As a result, the size will increase and migration will be retarded. So this results in a shift in mobility of this DNA. So this mechanism is called as electrophoretic mobility shift assay, and this is a very useful technique in studying interaction of a given DNA with proteins or other interacting partners.